And we'll move on to our last speaker of the night. It's Dr. Mike Andres. Mike is a research scientist at the University of Southern Mississippi, and he specializes in understanding how migratory fish and the parasites that call those fish home connect marine, estuarine, and freshwater habitats. He especially works with a Gulf sturgeon, which is a really cool fish, especially when you're up close and personal with them. So go ahead and take it away, Mike.
Yeah, thank you. Thanks to everyone. I didn't want to interfere too much with bedtime routine, but I'll start with my background. So I think I've had a strong interest in marine biology since the time I was in junior high. I grew up switching between Montana and the Seattle area. And so it was nice to experience a lot of outdoors and go into aquariums. But then I was really like moved by actually the Blue Planet series, and in particular the deep sea episode. So I got really interested in cephalopods. I started diving in the Pacific Northwest and seeing like the giant Pacific octopus. And that got me motivated to volunteer at the Seattle Aquarium. And I was given the opportunity to try to participate in a growth study where we were trying to look at growth rates of octopus in the wild versus those in captivity, which got me really excited. And so when I was looking for undergraduate institutions, I looked for some place that was marine oriented, but also away from Seattle. And so I settled on Texas A&M University at Galveston, not so much for the school, but because in Galveston there was an aquaculture facility dedicated for cephalopods. So I thought I could work with cephalopods at a lab and then do my undergrad research or studies in marine biology. I got there and I actually started working in a marine geology lab, just as some other way to keep doing research. And that never really stayed as a love for me, although it was exciting because those were my first two opportunities to participate in helping write and edit a manuscript for publication, which was a big change. My mind sort of exploded when I was an undergrad, and I was told that there's this entire phylum of organism that's very small, but they only live in the kidneys of demersal cephalopods, so cuttlefish and octopus, and they're considered to be a parasite, although we don't really know what their relationship is. And so that sort of totally shocked me that the thing that I thought I really enjoyed has this other thing that's really neat, the dicyemids. And so then I decided from that day on that I was going to pursue parasitology. So that led me to applying for grad school actually here with the University of Southern Mississippi. And for my PhD work, I worked on the taxonomy, systematics, and evolution of a group of parasites that lived in fish. And it was a great opportunity, as others have said, in terms of designing your own sort of research project, but it also provided a lot of opportunity for travel to Australia and throughout the Caribbean. So that was a lot of fun. And then when my PhD was over, I thought I had a nice postdoc that was lined up, and that fell through. So actually, I was sort of in between, and given an opportunity to work with someone else, Dr. Robert Leaf here at the research lab on menhaden. So I went from having this great idea that I was going to keep doing parasite evolutionary work, and then I started working on a forage fish, which fish have always been a strong interest of mine as well. In the summer times, I actually teach a marine ichthyology class. But that was a nice shift. And then after that, a different postdoc that actually involved working with Gulf sturgeon appeared. And so now in my current position as a research scientist, which fits somewhere in between being a research professor and being a postdoc, I've been given the opportunity to go back and apply some of my parasite work, but then work with a really neat fish. So hopefully I'm going to be able to share my screen and it'll work. So hopefully everybody's able to see these slides. I don't know why they're not fully pulled up, but the tags that I work with fall under sort of two main categories. Parasites under the biological tag. So we've got some parasitic crustaceans that are ectoparasites that actually came off Gulf sturgeon. And then there's these little black blobs are actually a trematode. So they're a flatworm that lives in the skin of this particular fish, a Spanish mackerel, that also makes migrations. And so try to use a couple of those as biological tags, and then we use some conventional tags like this Floy tag that you can sort of see over on the pectoral fin of the Gulf sturgeon, and acoustic telemetry tags. So the acoustic tag is being held by a technician ready for incision into the fish, and then we use I don't know if y'all can see my pointer, but then we use these passive acoustic telemetry arrays so we can track the movement of the fish. And so what you're going to see is fish that are juveniles, subadults, and adults, and their movement history over a 2-year pattern. So these fish are migratory. They move into freshwater each year and then move back out into the estuary and marine areas to feed, and they make pretty strong movement trajectories throughout their area. Which is important because Gulf sturgeon are federally listed as a threatened species, and in the state they're endangered. So it's been a lot of fun to work with them. I don't know how to stop sharing my screen. There we go. So I guess in my somewhat early stage, I think I'd like to echo what others have said in terms of being flexible, being a little bit open-minded, but working hard towards your end sort of goals and where you want to try to be. And then as in terms of, if you make it through all those stages and you're in the opportunity that most of us sit in, where you're pursuing funding opportunities, my dissertation advisor always said, follow the cheese. And so for me right now, it's actually been working on the conservation of a species that for the longest time I was honestly kind of bummed out that we weren't able to study the internal parasites of those fish. So it's kind of interesting. I went from cutting open a bunch of fish to look through their guts to now cutting them open and sewing them back up. So yeah, with that, I'll be happy to pause and take any questions you might have.
Thanks, Mike. All right, we got Ireland ready to ask questions.
Hey, thank you. So I actually have two questions, and one's kind of like a funny one, and then another one is like a serious one. So like, which one do you think we should have first?
Start funny.
Okay. So scientists are like very smart people, but they also like have a fun side. So do you have any like marine biology jokes that you can tell us?
I don't. No. This is sad, man. You're putting me on the spot with joke telling. No, I don't even have any bad like dad pun jokes for the marine world. I've been spending too much time working on the freshwater aspects of sturgeon, I guess.
Yeah.
I'm sorry.
I mean, there's always like the really sadistic jokes too, because of like global warming and stuff, but like those we don't have to listen to right now. But so my serious question is, so you work with like parasites and you learn about them. How are like fish populations as a whole being affected by like climate change? And like are parasites, are you seeing like an increase in like parasite deaths in like fish populations because of like warming of surface waters? Or
so the answer to that question is probably like every question, is it depends. So in some ways, yes, there are certain regions that are certainly more impacted by sort of range extensions of natural sort of native parasites into the other areas. But then also the situations where a fish is stressed and their immunity is not able to actually keep up. We tend to see this actually more associated with freshwater species necessarily than we sort of do with the marine species, or at least the marine ones are not well understood at this point. There's been some sort of anecdotal comments from colleagues in the Caribbean that actually surgeonfish, the blue tangs and doctorfish are starting to die for no apparent reason, and they're suspecting parasites might be the cause. There's colleagues that have been working on lionfish invasion for a while, and they sort of now have this mysterious thing that's popping up in introduced lionfish in the Atlantic, and they're blaming some form of parasite for it, not really understanding exactly what the mechanism is. I don't think that one fits into a warming perspective so much, but I think in the marine world we sort of lag behind freshwater type systems, for examples, just because of how open and how connected those systems tend to be versus the freshwater ones.
Thank you.
Yeah, sorry I fail on the joke.
I guess
that's a pretty good joke in the chat.
Thank you. I really tried.
All right, I think we have a question from Alice or Ilis. Clifford.
Thank you. So I'm from Bellevue, Washington, and I'm really interested in the Puget Sound and working with cephalopods. So I was wondering, what was the best part and the most challenging parts of doing that?
So where we lived was actually just right near you in Bothell. But I think the most difficult aspect was trying to control for factors in a invertebrate that we wanted to keep alive. So it would have been a lot easier to do the growth if we had more sacrificed individuals where we could look at sort of aging of the beaks along with the size, but that wasn't, their goal wasn't to kill their display organisms, right? So that was pretty challenging. I think Roland Anderson, so that's who I worked with. I think he's still there. He should be the curator of invertebrates as well at the Seattle Aquarium. But I think the most enjoyable aspect was actually being able to start as a high school student, getting introduced into some of the ways that you would carry out the research. So it was really shocking to me. I totally expected from when you looked at the front of the aquarium how clean and well maintained everything appeared, and then you got onto the backside and how it looked just as dirty as my kitchen sink does. It was pretty shocking. And then the way that some of those data records were maintained was also very, very shocking in terms of like their loose leaf papers that were just sort of all stacked and shuffling through them. So it was kind of a mess. So it was a good learning experience on how to try to take better notes and be more organized as well. All that though came, reflecting on much later in my career than then. Then I was kind of, a little bit peeved at them for being so poorly organized.
Awesome, Greg.
Thank you.
Thanks, Mike.
Yep.